Good day. Today we will be presenting our research on Chagas disease. This project is a collaborative effort. What is Chagas disease? Chagas disease, also known as American trypanosomiasis, is a potentially life-threatening illness caused by the protozoan parasite Trypanosoma cruzi. It remains a significant public health problem affecting millions of people primarily in Latin America. The parasite is mainly transmitted to humans through contact with the feces or urine of infected triatomine insects, commonly known as kissing bugs. These bugs often live in the cracks of poor quality housing. Transmission can also occur through contaminated food, blood transfusions, organ transplants, or from a mother to her baby during pregnancy. The infection progresses in two phases: an initial acute phase, which often has mild or no symptoms at all, and a chronic phase, which can develop over years and up to decades, and can lead to severe cardiac or digestive complications, or it can remain asymptomatic indefinitely. So, despite its severity, diagnosing Chagas disease faces several critical challenges. The current standard serological methods often require 2 or even 3 separate tests to get a conclusive result. This is not feasible for people in rural areas who may struggle to return to hospitals or clinics for follow-up visits, leading to many patients being lost due to follow-up. Furthermore, due to the high genetic diversity of the T. cruzi parasite across the different regions, individual tests often lack sufficient sensitivity and must be used in combination. Finally, these conventional tests are confined to laboratory settings and require trained personnel, making them inaccessible to many endemic communities. In response to the challenges that are faced when it comes to diagnosing Chagas disease, the aim of our research is to improve the diagnostic testing for Chagas disease by developing a single, affordable, and accessible rapid antibody diagnostic test. Our objectives is to achieve this aim by first designing a multi-epitope protein that can broadly detect antibodies against different strains of the parasite, and secondly, by using this protein to develop a rapid diagnostic test that improves the sensitivity, specificity, and speed of the current methods. Our methodology for our research is divided into two main parts. First, the design of the multi-epitope protein. So we select 3 well-characterized antigens: CAR AG1, CAR AG24, and TC24C4, and identify key antibody binding regions or epitopes from them. These epitopes are then going to be connected using flexible glycine-rich linkers to create a single chimeric protein sequence. This DNA sequence for this protein is synthesized and inserted into an expression vector with an affinity tag like a his tag. This vector is then introduced into an E. coli bacteria, which act as a factory to produce our recombinant protein. We then purify the protein and confirm its identity and purity using techniques such as the SDS page and Western blot. Finally, we will use the purified protein in an indirect ELISA test against serum from infected individuals to validate its effectiveness. The second part of our methodology involves developing the rapid diagnostic test itself. We will immobilize our newly validated multi-epitope protein onto a nitrocellulose membrane, which forms the test line of a lateral flow device. Simultaneously, we will conjugate anti-human immunoglobulin G and immunoglobulin M antibodies with a nanoparticle label, which will act as the detection agent. These components are then assembled into the final test device. When a patient's blood sample is applied, if antibodies against T. cruzi are present, they will bind to the labeled nanoparticles and then to the immobilized protein on the test line, producing a visible band. We anticipate from our research that the result of the project will be that our rapid diagnostic tests will provide a quick, accurate, and accessible diagnosis of Chagas disease in just one test. Because our multi-epitope protein includes targets from both acute and chronic phases of the disease, we expect the test to be capable of detecting infections at both stages, which is a significant improvement over some current tests. Based on the meta-analysis of existing rapid tests, we're targeting a sensitivity of at least 95% and a specificity of at least 97%. We also anticipate very little cross-reactivity with other diseases. This slide outlines the high-level budget allocation for our project. The majority of our funding will be dedicated to the research and development phases, including protein expression and purification, as well as the ELISA development and optimization, with our highest expense being the high-load 26600 SuperDex 75-page size chromatography, which will cost around 82,786 Rand and 48 cents. Our second highest expense would be the lateral flow assay kit, which would cost around 43,299 Rand and 75 cents. Significant resources are also allocated for the expanded validation and studies and the regulatory and manufacturing preparation required to bring a diagnostic test to the market. Our lowest expense being the SDS Page Gel Kit 10 assays, which would cost around 623 Rand and 52 cents. Our project timeline is structured to move systematically from design to dissemination. We will begin with the design and construction of the multi-epitope protein, followed by its expression and verification, and this is expected to take place in the first 4 months. Concurrently, we will then develop and optimize the indirect ELISA protocol. Once the protein is validated, we will design and create the first rapid test prototype. The middle stages, being month 7 all the way to month 16, will involve expanded laboratory validation and crucial field testing to gather user feedback. The final stages focus on navigating regulatory requirements, finalizing the test kit for manufacturing, publishing our results, and distributing the final product to stakeholders and health agencies.